Hello everyone. Today we're going to be talking about DNA. This is our introductory section for our gene action lesson. And don't be afraid to pause if you need to make any extra notes. I believe that most of you have these notes already filled in. So we'll hopefully be working with similar documents this time. So one of the things that we want to bring up here, this is going to be our most recent unit in terms of the development of science. We have already discussed genetics, which really owe their origins to like the 1800s and earlier. So there were some patterns discovered way early in the, even 600s and so about how certain traits would be inherited through a family. And then we discussed evolution, which is really a late 1800s, early 1900s concept. And from evolution, we went on to, I guess from evolution, we went on to discuss genetics and its sort of formalization, which all happened in those late 1800s as well. But DNA, RNA, protein synthesis, this is the newest stuff. This stuff comes out of the sort of 60s, 1960s and on, when really the good sort of technology came out. So like really good X-ray machines, good lab technique for discussing various compositions and other ideas like that. So prior to the 1800s, genetics was understood through its patterns, but not through the mechanism that allowed it to occur. Even sex-linked traits were known only on their unique pattern, not by the fact that they were linked to a particular chromosome. In the mid-1800s, rises of microbiology and biochemistry allowed for the discovery of cells, and quickly thereafter the tools to isolate different chemicals in those cells. Um, and then, it's really still like almost another 100 years before we really, like when I say mid-1800s, I'm talking like 1870 maybe. That's when really chemistry is starting to develop. And then it's not really until like 1930s, 1950s, where we really start to understand like the main building blocks that are going to be helpful here. So eventually we do discover that there are quite a few different important chemicals to the body. All cells essentially have a couple of different components that are really important. There's protein. These are chemicals that are made of amino acids. So that means that they're high in nitrogen. Amino acids are those things that you're always told you need to eat. There's the 20 something amino acids, the essential amino acids you got to eat. You get those from eating meats and proteins from high protein plants. Some of these amino acids you're required to get from your diet. Some amino acids your body can make. And then they put those together and they make those into proteins. And those proteins are really what's responsible for the chemical reactions in the cell. Protein is what makes your cell go. I'm going to put that in here. So these make the cell go. Okay? They're the engines, they're the gears, they're even the structural components. They are the, when we think about even building a building, these would be they would be equivalent to like the brackets and the bolts that hold parts of the building together. They're as important as the hinge on a door, right? Have a hinge, and those hinges would be proteins in the cells. There are some structural pieces like fats, lipids, and phospholipids. In terms of a building, fats, lipids, and phospholipids might be like building materials like like wood. Wood is kind of like a phospholipid in a way. Like the wood beams that might make up a build. It might be there for the bones of a building. Even like the gyprock that makes up your walls, or the plaster. That stuff might be the equivalent of fats and lipids in your body. If you are a car, fats and lipids might be a good representation might be like the gas in the car. Sure, you got gas in the car, but it can't do anything without all the engine parts, and all the engine parts would be equivalent to the protein. So, every single moving part in a cell is a protein. Every single stationary parts and storage parts are equivalent. So almost all. I'll give that because I'm a little worried that someone's going to call me out and say, oh yeah, but what about these? And I'll be like, oh, okay, yeah, you're right. But almost all the moving parts of the cell, right, because DNA is one of the moving parts that is not a protein, are either proteins or able to move because of a protein. Oh, this is a weird. I've never had it jump up and down like that. Okay. Can I convert the text? These make the cell go. Almost all the moving parts of the cell are either proteins or able to move because of the protein, able to function. Function in the cell is really always driven by proteins. Essentially almost always driven by proteins. Fats, lipids, and phospholipids, these are really important chemicals too, but they are mostly for structure. And they're mostly made of carbon and some phosphorus. And I put that in there because I want to reference back to the chemical compositions later, as some of the early studies on how did the cell function, how does inheritance work, was really based on watching for some of these chemical markers. Nucleic acids is the third chemical that's important to the cell. And it is the least amount of the cell. It's a very small portion of the cell. I believe it's less than 5% of the cell by mass is made of nucleic acids. So it's a very small part. So this is the smallest component, or smallest proportion of the cell. But it's made of phosphorus and sugars and nitrogen bases, and there are also some protein that's usually tied into that. And they come in two different types. So there's two different types of this building material. There's the ribonucleic acid, and there's the deoxyribonucleic acid. And the other name is the shorthand RNA and DNA. So a nucleic acid is a generic type of chemical that's found in the cell, and that contains sugar, nitrogen, and phosphorus. It's connected in long chemical strands, which the chemists would call a polymer. Polymer strands, and it is made of two different types of chemical. It comes in two different versions: the RNA or the DNA version. And the RNA version was discovered first because there's usually more RNA floating around in the outside of the cell. Meanwhile, the DNA was a little bit harder to find. It's found as particularly inside of the nucleus. So after the 1800s and into the early 1900s, we have this idea that the cell is made of these three different components. And there is an interest in, okay, well, we know that all cells come from other cells. And we know all organisms are made of cells, and all organisms were made of cells with organelles. We knew that chromosomes were likely related to genetics, as they were visible as a part of meiosis and mitosis. And we also had an idea that chromosomes could form chromatin in the nucleus, but we didn't know if inheritance was directly related to fats. Was it directly that were a large portion of the cell? Was it related to proteins that were found in all cells? Or was it related to nucleic acid, a little tiny portion of the cell, but found in almost all the types of cells, but not all cells, right? So even like red blood cells don't have nucleic acid. So, or they don't have, at least they don't have DNA because they don't have a nucleus. So there was some question about how important could nucleic acids be when they're such a small portion of the cell. Now we already should know the answer to this because we've already used this language before, and we've already talked about DNA as a unit of inheritance, but we are going backwards in time, imagining a time when you wouldn't have known that. And how would scientists have worked their way around this thinking that there was these different parts of the cell and which one was important to reproduction? So we're going to go quickly through a couple of examples of the history, and then we will go into some history. We'll go through the history of how we decided on nucleic acid being important, and then in the next video we'll talk about how the structure of the DNA was discovered. So the first important person to talk about is Friedrich Miescher, is how I would probably pronounce it. So he was working on pus from bandages, and later he worked on sperm from salmon. These are both chemicals that have cells with lots of genetic information in them. White blood cells have multiple nuclei, and so they are carrying a lot of chromosomes. So it just so happens that because this scientist decided to work on this particular type of cell, he ended up working with a lot of chromosomes, whether he knew it or not. And so he discovered this organic chemical that had a high level of phosphorus, and he didn't know what it was. But he figured it must have been from the nucleus because the cells he was working with had nucleuses in them, pus particularly, and so he called it nuclein. Comes from the nucleus. This chemical probably comes from the nucleus, so let's call it nuclein, and it is a phosphorus protein chemical. Very quickly, scientists started to think about nuclein and chromosomes and think, well, maybe these are the same thing. If this is a chemical coming from the nucleus, it's very hard to figure out where chemicals are coming from when you're using a sort of basic materials, basic equipment in the 1800s, but possibly this is actually the chromosomes that he's detecting. So Frederick Griffith was interested in discovering whether this chromatin material, or whether whether one of these three chemicals was important to inheritance. So this is a really interesting experiment that he used. Here is how it works. He takes a living strain of pneumonia, a staphylococcus chemical, or a streptococcus. So streptococcus is strep throat. So in certain types of strep throat can cause pneumonia. This type is called Streptococcus pneumonia. So this is a type of bacteria that can cause pneumonia in rats, and probably in people as well. And so he took this strain, and he had one strain that was a viral strain. So this means that the strain would cause this would be dangerous to the mouse. So injecting the viral strain into a mouse would cause it to die. And he could even take cells out of the heart, and he could find these bacterial cells in the heart that he could grow on a plate, and he could identify them as the viral strain. Now he also had a strain of Streptococcus that was not viral. So this type here, injecting this type into a mouse, there would be no effect, and the bacteria would not spread to the heart, would not cause death. So he decides to take the original viral strain, right? The very original dangerous strain. He moves it over a flame and kills the bacteria. Bacteria dead. And he injects that mixture into the mouse, and it's healthy because, of course, the bacteria are dead, so they are not able to cause damage to the mouse. Then he mixes the two together. He mixes the dead viral cells and the non-dangerous cells, the living but not viral cells. Mixes them together, and suddenly the mouse dies again, and living S strain was found in the heart. The same effect as when the mouse was affected by the living S strain. So this suggested that the transfer of genetic information does not require life. He essentially proposed that the dead S strain could transfer its information to the living R strain, and that R strain could take in this information and convert it into a new trait, a dangerous trait that would make that strain become the S strain. So this is a property that bacteria, we learned a little bit about this when we talked about bacteria back in the day, that bacteria, when they are required, when they're in danger, they can even create these little clusters that store their genetic information, and those clusters can be picked up by the next generation of that bacteria to create, to gain those traits. So this is a way of horizontal gene transfer. But this is the first time that this is being observed, that life is not that traits that give life forms their abilities, their genetic functions, those traits do not require a living component. So now the search is on. Let's look for some chemical that can cause this. Oswald and Avery do a really exciting version of this experiment. They decide, okay, well, now we know that this dead material can cause damage. Let's use some chemicals to break down the different components inside that mixture. So they take the type 3S, so here it is, the S bacterial filtrate, so that's the virulent. This is virulent, right? Virulent, this is the dead virulent cells. So Oswald and Avery take some dead virulent cells, and then they apply different enzymes to them, and before they mix it with that R cell. And then they observe that same effect. Does this mixture now have the ability to kill a mouse or not? And so the first time they apply RNase, which is a chemical that specifically targets RNA and destroys it. They're not really interested in fats in this experiment because fats usually break down under heat anyways. So the heat treatment immediately discounted the lipids, which are broken down. Fats. And so they apply this RNase, it breaks down that RNA, and they still observe that the bacteria becomes dangerous. They observe both types of the bacteria. The bacteria is able to convert to the S type. They destroy the protein in another experiment, but they don't destroy the RNA and the DNA. Just like in the first experiment, they don't destroy the RNA and the DNA. They don't destroy the DNA. And they still see conversion. In the final experiment, they use DNase. DNase is a chemical that destroys DNA. So now this solution only contains protein and RNA. It does not contain DNA anymore. It's been destroyed. Now there is no change. The bacteria stays as the R type. So this filtrate loses its ability to convert R type into S type when you destroy the DNA. This implies that DNA is the mechanism by which the R type is being converted to the S type. DNA is the chemical unit that is carrying the information necessary for the R type to turn into S type. DNA is essential to the to the inheritance of the R-type, of the S trait in the R-type bacteria. Hershey and Chase do a very similar experiment. They really want to get down to make sure that it's not protein, because DNA does have some protein in it. Maybe that's what they're concerned about. So they decide that they're going to do this experiment with radiation. They find a way that they can apply a radiation label to protein, or apply a different radiation label to DNA. And they grow some virus with the different types of radioactive label. When the radioactive label is placed on the protein, the virus attacks a bacterial cell, and the bacterial cell stays not radioactive. So there's no transfer of radioactivity when they make the proteins radioactive. The bacteria still becomes infected with the virus, but the bacteria does not show any radioactivity. So it still gains the trait. So it gains the trait, gains the infection, but not the radioactivity. And meanwhile, in the second experiment, they label the DNA, and the DNA label goes in and they find that the infection by the virus causes the bacteria. So the bacteria becomes infected, and radioactive. So that suggests that the DNA is the tool. The DNA is the unit that is carrying this this trait. So when the DNA is made radioactive, not only is the trait of infection carried, but also the radioactive property. So I'm going to leave it there for now, and we'll come back and talk about some of the essential discoveries that led to the structure of DNA in our next lesson. Have yourselves a great day. We'll talk to you soon.